again, you will not be asked direct questions about the metabolism, لكن as you will see later, and we will be talking إنه إذا كان على سبيل المثال that specific structural modification was done to overcome that metabolism اللي بيقلل من half time and so on. But at a certain point, يعني مثلا لما ينحكي إنه بصير في عندي على سبيل المثال OD alkylation followed by let's say phase 2 metabolism or conjugation, so you need to familiarize yourself about these concepts. لازم تكونوا فاهمينها، مش إنه حافظينها 100%. مش راح تنسألوا direct unless إذا إحنا ناقشنا بشكل extensive عن certain drugs الميتابوليزم ممكن تنسألوا عنها مباشرة، بس إنه what do you expect الميتابوليزم هالدرج إيش راح يكون زي ما أخذتوها مثلا بالميديسينال 1 أو بالأورجانيك لا. alright. فهلا those two slides alright somehow summarizes everything you have covered بالميتابوليزم اللي أخذتوه تقريبا for almost a month بمادة الأورجانيك.
دكتورة.
طيب. yes.
يعطيك العافية.
الله يعافيك.
في طلاب بيحاولوا يدخلوا على المحاضرة بس بيحكوا إنه أنت مو قابلتهم لسه.
طيب.
عشان بس يقدروا يدخلوا على المحاضرة.
alright. يعني I was hoping إنه لو عملنا هالشيء أو عملتوا هالشيء قبل. لا ما في أي حدا let's say موجود requesting joining. هلا في لينك موجودة على e-learning يعني through it you can let's say join in the team. معلش أغلبكم يعني share this. طيب. هلا هاي الانترودكشن بسيطة عن الكانسر for you to read, just let's say to refresh your memory about it. بس إنه تنسألوا مثلا what is the definition of cancer, what are the types of cancer, the terminology used and so on. أكيد هاي كمان it's not the interest of us as medicinal chemists. أنا فقط اللي بدي أحكيها as an introduction. وبعدين we will start with the first class of antineoplastics. إنه even though يعني there is let's say tremendous خلينا نحكي خلينا نحكي advancement في الانتي كانسر chemotherapy let's say research a huge number of anti-cancers being FDA approved بالadvancement بالtreatments للكانسر لكن still a number of cases to be diagnosed is expected to let's say almost double 1.5 times by the year of 2040. يعني تقريبا متوقع انه يتضاعف الكيسز اوف كانسر being diagnosed تقريبا مره ونص بسنه 2040 وبرضه كمان رح تتضاعف اعداد الوفيات بغض النظر عن الايرلي ديتكشن اللي عم بصير او الادفانسمنت بالايرلي ديتكشن وبالتريتمنت and so on. Mostly because the anti-cancers are facing resistance. يعني في كثير patients للاسف يعني ممكن هالتريتمنت بينفع معهم يعني بعد فتره بسيطه it may not work أو it fails بسبب الريزيستنس اللي that cancerous cells can develop to resist the effect of that anti-cancer. فده خلينا نحكي the main reason why there is let's say أو it is expected in the cancer cases being diagnosed the deaths due to cancer are expected to increase. Alright? فعشان هيك دائما في continuous research and search for new anti-cancers targeting new targets, new signaling pathways, and so on. تقريبا a number of anti-cancers being approved by DNA or those let's say investigational drugs that are filed to the FDA to treat cancer مقارنة ب any other drugs to treat any other disease is way much more. Alright? فعشان هيك what we يعني لو مثلا بدكم ترجعوا للليترشر رح تشوفوا إنه معظم الأبحاث اللي عم بتصير is to investigate new targets to treat cancer, to develop and design new drugs for treating cancer simply لأنه كثير من التريتمنتس اللي already موجودة for many patients are failing. طيب هلا أول class رح نحكي عنه اللي هم those affecting the DNA. وان particular راح نبلش باول كلاس اللي هم الانتركاليتنج drugs. فهلا قبل ما نناقش انه كيف هاي drugs do interact with DNA, I need, let's say, to make sure that you already know what we mean by DNA, what is its structure, ايش ال expected interactions اللي ممكن الدراجز تعملها مع ال DNA. So, what is DNA? It's a polymer. Polymer of what? Nucleotides. And the nucleotides are made of what? No? Yes. Yes. Yeah. So it's a phosphate group. All right, a sugar unit, ribose or deoxyribose, depending on can DNA or RNA, and for sure nucleic bases. How many nucleic bases do we have? Oh, let's say yes, 5. All right. طيب هلا what you see in here is a strand, a single strand of DNA that is composed of, as we have mentioned, the phosphate group, which is depicted in here as this yellow circle. Tamam? This is the sugar part, and this is where the nucleic base is. The nucleic bases are classified into pyrimidines and purines. The pyrimidines اللي هم cytosine, thymine, and uracil. طبعا في DNA, the nucleic bases that you see are thymine and cytosine. Instead of thymine in RNA, you see uracil. And as for the purines, which are actually 2 fused rings together, اللي هم الأدينين والجوانين, you see them both in DNA and RNA. تمام? Now, as for the sugar part, in DNA, it's deoxyribose. In RNA, it's ribose. All right? هلا deoxyribose versus ribose, because at position number, let's say, yes, 2, instead of an OH, you have an OH. طيب. Now, we all know that DNA does not exist as a single strand, وإنما 2 strands, all right, or a double strand that are, or that form, what is the secondary structure for it? It's a helix or a double helix, okay? أو بمعنى آخر, it has a helical structure. طيب, هلا depending على the space ما بين هاي strands, if it's in يعني ب one turn, the space between one strand and the other, that is called a minor groove. Between one turn and the second turn, that space in between them, we call it a major groove. This is important لأنه later رح نحكي عن some antineoplastics, they do approach the DNA or they interact with the DNA from the minor groove or the major groove. So this is something important for you to understand. طيب, هلا going back to the nucleic bases. Sorry. هلا if you look at, all right. If you look at, for instance, this double helix or this double-stranded DNA, the space in between one helix and the other, what we mean by helix, it has, let's say, a loop-like structure, or you have turns in the structure of the DNA strand or double strand. The space between one strand and the other, بنفس turn, all right, هاي بنسميها احنا minor groove. Between one turn and the one that is next to it, or one turn and the second turn, that space is called a major groove. For sure, as you can see, the space in the major groove, as the name indicates, is much bigger than the one in the minor groove. طيب. هلا, what are the bonds that holds the 2 helices together? Oh, as the 2 strands in the helix together. Hydrogen bond interactions ما بين إيش وإيش? Between the nucleic bases that are, for, let's say, in between each other. طيب, هلا these, let's say, circles, okay, these cyclic structures which indicate the nucleic bases, the adenine, the guanine, the cytosine with thymine, as you can see, it's drawn as if it's planar, right? Why is it planar? Let's go back to this structure. Why is it planar? What is the if you look, for instance, at cytosine, what is the hybridization of all of the atoms comprising this structure? كلهم sp2 واحنا بنعرف انه ال sp2 hybridized carbon او atoms, all right, they have a planar geometry. Being all sp2 hybridized معناها انه كل هاي الatoms that comprise that ring system are actually planar in geometry. So that is why if you look at this structure, all of those nucleic bases are drawn as if they are all on the same plane. All right? هلا the dots that you see here represents اللي هم يعني let me go back to for instance الأدينين على سبيل المثال. This nitrogen here. All right? So that nitrogen can form hydrogen bond interaction with the amino group that is in thymine, as you can see, let's say here. The oxygen will even in nitrogen with 2 oxygens. All right, we will go back into more detail بس نحكي عن الالكيليتنج agents. راح احكي عن هاي الانتراكشنز بشكل even more detailed لانها very important حتى كمان نفهم ليش مثلا alkylating agents يعتبروا cytotoxic. We will talk about that again in more detail. هلا how many hydrogen bond can be formed between highly nucleic bases? 3 or 2 depending على البيس pairs. If it's ما بين adenine and thymine, 2 hydrogen bonds, guanine and cytosine, 3 hydrogen bonds. طيب, is this the only interaction that binds the 2 helices or acid, the 2 strands in a DNA helix together? Yes. Which is but this is a covalent bond. أنا بحكي عن non-covalent interactions. يعني what you are saying ما بين الphosphate مثلا with ribose with other phosphate and so on, these are covalent bonds, very very strong interactions. This is what keeps, let's say, the bonds, or what keeps, let's say, one strand for sure. These are very 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 strong bonds. أنا اللي عم بسأل عنه اللي هي الانتراكشنز ما بين, let's say, 2 strands أو أسفل, yeah, 2 strands بالhelix together. Besides the hydrogen bond interaction, there's also a very important interaction. Disulfide, لا, ما تحتاج, we're talking about 2 sulfurs, let's say, adjacent together. ايش هي عفوا؟ Van der Waals interaction, or to be more specific اللي احنا بنسميها ال pi-pi stacking. هلا between, let's say, in each base pair, what is holding them together اللي هي to keep the 2 strands held together. In between the base pairs and the one that is, let's say, right next to it, so what can below it or above it, those, let's say, rings are also held together by what is called pi-pi stacking. Okay? Oh, much weaker than hydrogen bonding, like, and still it's very very crucial for binding DNA strands together in a helix. طيب. هلا the 2 figures you see in here represent the DNA as if I am looking at it directly, or if you rotate it and you look at this second image in here, as if you are looking at the DNA from a different angle or a 90-degree angle versus this one. The one that is in orange represents what you think? Sugar backbone, all right? اللي هو phosphates bound to sugar. What about the one in purple? البيس pairs, okay? هلا اطلعوا هون. لو أنا بدي أطلع, أو if you look at this image, هلا البيس pairs versus the backbone, مين more lipophilic? Which is more lipophilic? الباكبون ولا النيوكليك bases in the center? Yeah, exactly. Yeah, it's way much more lipophilic. The backbone, which is composed of ribose and phosphates, the phosphates, it's an ionizable group, راح يكون في عليهم negative charge. For that part of the molecule, as you can see, يعني تطلعوا هنا كيف موجودة, as if وكأنه أنا عندي the backbone is surrounding the base pairs اللي موجودة في نص ال DNA. What is the media in which the DNA is present in? بالnucleus. الميديا. Yeah, for sure الcytoplasm أو let's say الcytosol. What is it made of? ايش الميجر solvent الموجود؟ ميه. Exactly. But they are present in here in the lipophilic part وكأنه hidden away from the water. Will backbone of the DNA, which is ionizable, which is highly polar, is the one that is facing or confronting the water, forming اللي هي interactions with it. تمام؟ All right. Here's another, let's say, image just to show you اللي هي major group and minor group. So in each term, the space ما بين one strand with other strand, that's called the minor group. Between one term and the one, let's say, next to it, the space in between, that's called the major group. طيب نبلش هلا نحكي عن الانتركاليتنج agents. هلا اللي عملته في هالسلايد انه انا جمعتهم كلهم مع بعض so that you will be able to distinguish ايش هي major parts that makes an intercalating agent. هي هم نفسهم اللي موجودين عندكم يعني ف it will be confusing for you يعني هي انا حطيتهم كلهم مع بعض. طبعا we will be discussing each one by its own هلا بس انه انا حطيتهم to ask you a few questions على اساس انه نرسخ فكره what we mean by intercalating agents. Okay, these are molecules that interact with DNA. طيب اطلعوا عليهم كلهم. Those intercalating agents موجودين عندكم كل واحد لحاله. What I did in this slide, I just, let's say, gathered them على أساس تعرفوا إيش الإشي المشترك بينهم. ليش أنا بسمي هاي intercalating agents as such. طيب, look at them. All of those 5 intercalating agents that we will be discussing. إيش الإشي اللي بيجمع بينهم؟ إيش الإشي common between all of them structure-wise؟ كلهم planar. يعني مثلا لما أطلع على كل واحد فيهم كلياته يعني كل parts of it planar. Yeah. So in all of them, as you can see, all right, they do have a ring system that is planar. تمام؟ طيب bleomycin, which part you think may resemble a part that is planar? Yeah, those 2 rings in here. هي بيسموها thiazole. So this is what combines all of those intercalating agents that we will be discussing. They do have a part in their structure that is planar, a cyclic structure. So it can, let's say, fused. So it can, let's say, linked together. So attached, let's say, together directly. Or composed of 2 ring systems or 3 ring systems. What combines between all of those parts of hermacophores and those intercalating agents is that they are planar in structure. Okay. طيب هلا كل هاي الرينجز planar. For sure, those 2, let's say, rings in here definitely planar. What about this one in here? Do you think إنه هاي الatoms راح تكون برضو planar with those 2 adjacent rings? What do you think? What is the hybridization of this oxygen? sp2. All right. What about this nitrogen? برضو كمان sp2. So زيهم زي all of those carbons that constitute those 2 rings, they are all planar. تمام؟ طيب what else? ايش في شيء ثاني ممكن let's say يكون خلينا نحكي هيك common between them. High molecular weight مش كلهم يعني for instance amsaccharin sorry is not يعني it doesn't have a huge molecular weight. Yeah, so in many of them you will see for instance طبعا مش فيهم كلهم, it's not a must, but على سبيل المثال بالdoxorubicin for instance بالmitoxantrone, those 2 drugs they do have an aliphatic nitrogen that will be ionizable. ورح نحكي later on إنه what is the importance of that group. هلا in the rest of them طبعا aside from bleomycin رح نحكي عنه كمان بتفصيل أكثر later on. We do have in the let's say planar part of those drugs, you will see now we have many groups that are highly polar that can form strong hydrogen bond interactions. All right. هلا what constitutes or what makes, let's say, those agents or those drugs أو ليش احنا بنسميهم intercalating agents, because they can simply the planar part, and recall the DNA structure we do have base pairs that are parallel to each other, right? هلا intercalating agents, what they do is that they insert the planar part between high adjacent base pairs, and they form interactions with the base pairs above and below. And as for the part which is non-planar, all right, they do form either ionic interactions or hydrogen bonds, strong hydrogen bond interactions مع خلينا نحكي الباكبون, let's say, of the DNA. و not only the backbone, برضه كمان ممكن يعملوا interactions مع البيس pairs. We will talk about that in راح نفصل كل drug ايش الانتراكشنز اللي بيعملها. All right, here's another, let's say, animation which shows you the strand of the DNA. All right, and as you can see, these are the base pairs in DNA structure. This a planar part of the intercalating agent, it inserts itself between adjacent base pairs. And as for the either ionizable part of it, the part that can be ionized at physiological pH, or the part of the intercalating agent that is considered highly polar, it can form either ionic interactions مع الباكبون سواء كان الفوسفات أو حتى الريبوز, أو ممكن يعمل, let's say, strong hydrogen bond interaction. Okay. هلا أول دراج رح نحكي عنه اللي هو الدكتينومايسين. طب do you expect that this is a drug that was synthesized in lab? What do you think? Do you think, this is a drug that can be easily synthesized in lab, or you think it was isolated from nature? Yeah, that's let's say a natural compound isolated from nature. طبعا, and you can judge that by looking at the complexity of its structure. هلا مرسوم هون بطريقة مختلفة عن اللي حطيته بأول سلايد. Okay. هون instead I have val, pro, val, gly. What do those indicate? Amino acids. So attached to this planar part of this drug, okay, it is bound to two cyclic structures which are composed of amino acids. Okay. Val stands for what? Valine. The side chain of valine. Do you remember what it is? Does it have any? لا, valine لا. Yeah, it's an isopropyl group. Okay. The glycine. Yes, there's no, the side chain is actually a hydrogen. What about proline? Yeah, it's a ring structure that connects the nitrogen to the, let's say, the alpha carbon to the nitrogen. تمام؟ طيب. هلا, this nitrogen in here, in dactinomycin, is it ionizable at physiological pH? شو رأيكم؟ يعني, would you expect the dactinomycin to form ionic interactions مع DNA? لا. لا. ليه? What do you expect the pKa for it to be? Around 10, 11 زي الأليفاتيك amines مثلا? Yeah, أقل. ليش طيب؟ Exactly, because the lone pairs on this nitrogen are not localized على nitrogen, وإنما they are delocalized, let's say, with this system in here. That is why they are highly withdrawn from the nitrogen. Electron density عليها جدا قليلة. ال pKa راح تكون واطية, يعني 5 وحتى أقل من 5. So you would not expect this nitrogen to be, let's say, ionized at physiological pH. All right? This compound does not form ionic interactions مع DNA, وإنما it only forms hydrogen bond. يعني this nitrogen, if you would be asked about the type of interactions it can form with the DNA, for sure راح تكون عندي hydrogen bond interactions. All right. طيب. هلا. The image that you see in here, and هلا بنشوف كمان crystal structure for it. The image you see in here is the DNA and the actinomycin. This is an animation of its structure. The green part, you think it represents what? The planar part of it, and those two huge, let's say, spherical shapes represent those two, let's say, cyclic peptides. All right. Now, the approaches the DNA from the minor groove. That is why it was important for us to understand what we mean by minor groove and major groove. Okay. طيب خلينا نشوف هلا a crystal structure of this drug. I will show you two actually. I need you to extract what interactions this drug forms. أنا حكيت لكم من المحاضرة الماضية إنه كثير راح أعتمد بالذات بالأنتينيوبلاستكس وحتى كمان يعني بكل السبجكتس اللي راح نناقشها كثير راح نعتمد على ال PDB database. يعني it has very nice crystal structures of our targeted drugs with the biomolecules they bind to. فهي كثير بتسهل علينا نفهم الانتراكشنز، نفهم ايش هي الفانكشناليتيز اللي جدا ضروريه. يعني for instance بحاله مثلا الدكتينومايسين اي change بعمله بحيث انه بغير البلاناريتي of the planar ring بتوقع انه yeah the activity will be highly, let's say, reduced او حتى ممكن تكون diminished. برضه كمان ال functionalities that are important بالcyclic pentapeptides, any change in it بالذات الفانكشناليتيز اللي بتعمل interactions بتوقع بهاي الحاله انه الاكتيفيتي will be diminished in that manner too. طيب. So what you see in here, okay, represents the structure of DNA bound to actinomycin. Again, that green part you see in here represents the planar part. And as you can see, those two green structures in here represent the cyclic peptide. طيب اللي معاه notes just يعني خلوا البيج على الستركتشر عشان بدي أسألكم عن مثلا إيش الفانكشناليتيز اللي ممكن تعمل interactions. طيب هلا another thing that is written in your slides إنه the actinomycin favors binding to the DNA where guanine and cytosine base pairs are. Okay. هلا why it favors that? Why the actinomycin لما يرتبط بيكون أحسن ارتباط له إنه to insert itself between guanine cytosine base pairs لأنه بيعمل interactions معها. All right. طيب let's zoom in now على ال planar part in here. معلش الماوس حركته عكسية. طيب. So this is the planar part of the actinomycin or actinomycin D, and both are synonyms of the same drug. These are, let's say, the base pairs of the DNA between which the planar part inserts itself. طيب هلا usually بهاي crystal structures interactions أو intramolecular أو inter أو حتى intra molecular interactions, the non-covalent interactions are represented by dashed lines. Okay, فهاي dashed lines اللي شايفينها هون represents non-covalent interactions اللي بيعملها التارجت مع الدرug. Alright. طيب. هلا these parts in here أو this let's say group in here لو بدكم تقارنوا اللي موجود هون بالستركتشر اللي مرسوم عندكم. What does this nitrogen refer to? Oh, sorry. It's a group in here. Yeah, it refers to the nitrogen. يعني بهالكريستال ستراكشر I will show you another one بتكون الانتراكشنز كثير اوضح هلا من وضحه. بس حبيت افرجيكم let's say a crystal that is less او crystal structure that is less crowded على اساس نشوف اللي هي some of the interactions مش كلهم اكيد. طيب هلا مكتوب عندكم في السلايدز انه the reason why this drug favors الجوانين cytosine intercalation لأنه الجوانين in particular the two nitrogens of the guanine, okay, let me zoom in عليها, the two nitrogens of the guanine in here, that base pair form interactions, you think with what? شو رأيكم؟ بما إنه this is the planar part and these are the cyclic pentapeptides, مع إيش بيعمل interactions؟ Yes, that amide which links the cyclic pentapeptide with the planar part. Okay, for any change بهالstructure being part of the structure for sure will affect the activity. طيب خليني أرجع ل another crystal structure واللي بيكون فيه interactions أوضح. Okay. So this is a crystal structure of actinomycin D or actinomycin with DNA. All right. هلا DNA is shown in orange. هاي الred dots اللي شايفين هون أو orange dots these represent the water molecules. All right. هلا what you see in here this represents the backbone of DNA. These are the nucleic bases. ولاحظوا إنه أنا عندي as we have mentioned the planar part in here is what inserts itself between the base pairs. All right. And this is these are the two cyclic pentapeptides. If you click على the drug structure, it will show you instantly the interactions اللي بيعملها. Okay. Response تبقى شوي بطيء. All right. لاحظوا إنه أنا في عندي هاي the green dashed lines between this planar part والcytosine والguanine. تتوقعوا إيش هاي الانتراكشنز؟ No. No. Pi stacking, exactly. يعني زي ما base pairs they can form pi stacking between each other. برضو كمان that planar part of the drug when it inserts itself between the base pairs it can form pi stacking with the base pairs on top and below. تمام؟ Okay. أنا مش هحكي عن كل الانتراكشنز ومش مطلوب منكم تعرفوا everything about the interactions that the drug forms بالذات لما نحكي عن very complex structures. But at least what is important for activity, this is what we need, let's say, to identify. هلا حكينا إنه النيتروجين in the planar part. Yeah. معلش تحملوني شوي وأنا بحرك فيها. This nitrogen in here in the planar part لاحظوا إنها عاملة interactions مع إيش؟ Let me zoom in. With what? What does this line represent? Yeah, the backbone with the oxygens of the, let's say, the phosphate and the oxygen of the deoxyribose. That nitrogen can form interactions with them. Are these ionic interactions? Yeah, مش ionic لأنه إحنا already بنعرف إنه هاي nitrogen is not ionized at physiological pH. The kind of interaction that they form are actually hydrogen bond interactions. This is very crucial. إذا شلت النيتروجين غيرتها بشيء that cannot form hydrogen bond interaction, I will be losing in that matter the activity. Okay. هلا one last interaction بدي أفرجيكم إياه اللي هو just bear with me معلش. that base pair in here, which is adenine. That adenine, the 2 nitrogens of adenine, let me see if I can say zoom in. The 2 nitrogens of adenine in here, as guanine. The 2 nitrogens of guanine, they form interactions with the nitrogen and oxygen of the amide bond that links the planar part with cyclic structure. Okay? Well, these are not the only interactions. برضو كمان the cyclic structures they interact مع الباكبون مع النيوكليك bases اللي موجودين عندي في الماينر جروب. alright. طيب.
دكتورة ما في صوت.
هيك سامعيني يا جماعة؟ يس.
سامعينك دكتورة.
طيب. So again, let me repeat the question. هلا how do you think the cytotoxicity of those drugs بالذات على الcancerous cells results from when they bind to DNA? يعني هل مجرد binding of that set this will result eventually in the death of those cancerous cells? Yeah. So they for sure not only they will stabilize DNA, they will prevent the replication, they will prevent transcription. So those enzymes which are involved, all right, in replication and in transcription of DNA, they will be inhibited, right? What is known about الدactinomycin or actinomycin, when it binds to DNA, it also stabilizes a complex of DNA with the enzyme that is responsible for RNA synthesis. هلا شو بتتوقعوا الانزيم؟ بس هيك بدي اساعدكم عشان تحفظوا اسمه. What do you expect the name of the enzyme that will synthesize the RNA شو راح يكون اسمه؟ Bravo. It's RNA polymerase for sure. And because it relies on a DNA template to synthesize the RNA, it's called DNA-dependent RNA polymerase. طبعا مش بس this is not يعني اللي موجود عندكم بالسلايدز إنه this is the major enzyme that is خلينا نحكي inhibited or poisoned. راح أحكي later بالمحاضرة الجاية إن شاء الله what we mean by or what is the difference between an inhibitor and a poison. It's not the only enzyme that is poisoned by those, let's say, intercalating, or this specific intercalating agent. برضو كمان, literature is showing that it can also inhibit topoisomerase 2. رح نحكي عنه بتفصيل, topoisomerase 2. التفصيل عشان بس نفهم what it does and how the later drugs inhibit it. بس إنه as a, خلينا نحكي molecular biology information, مش رح تنسألوا عنها مباشرة في الامتحان. All right. Now, what is known again about the actinomycin D is that it stabilizes not only DNA structure, but also the DNA structure when it complexes with DNA-dependent RNA polymerase. This is an animation of this enzyme where it shows you that this enzyme, when it binds to the DNA, it uses one of the helices of DNA as a template to synthesize the RNA, which is shown here in red. So when actinomycin D binds to DNA, it also can bind very well. يعني برضو كمان رح يعمل interactions مع ال DNA-dependent RNA polymerase, and it will poison that enzyme, stalling its function. وبالتالي it will prevent the transcription. Will also بما إنه كمان ممكن يعمل inhibition لtopoisomerase 2. It can also inhibit DNA replication, and all of those خلينا نحكي mechanisms will eventually lead to apoptosis or killing of those cancerous cells. For sure, in normal cells can be also affected, بس لأنه cancerous cells they have a much higher rapid or much higher rate of DNA replication and DNA transcription. That is why they are affected, let's say, much more. Okay. طيب. هلا ننتقل ل another intercalating agent اللي هو الدوكسيروبيسين. Look at this part واحكوا لي which ring system or which part of it is the one that will intercalate with the DNA. يعني أنا عندي 1, 2, 3, 4, 5 rings. Which ring or which rings you think will intercalate with the DNA? 1, 2, and 3. What about this one? Why it's not planar? Yeah, because we have, let's say, sp3 hybridized carbon. For sure, if you would look at the geometry of this ring, it's definitely not planar. So it's not expected that part of the drug, this part in here, those two rings will for sure will not intercalate with the DNA. That planar part is the one that will be inserting itself between base pairs, the DNA structure. Okay, fine. Do you have any ionizable group in the doxorubicin structure? Yeah, the nitrogen in here, all right, yes, is the one that will be ionized at physiological pH. شو بتتوقع ال pKa for it will be? Yeah, around 10, something like that. Being around 10 معناها physiological pH for it is acidic media, but definitely it will be mostly at a very, very high percent, it will be ionized at physiological pH. But I would expect زي ما رح نشوف later on be, let's say, crystal structure for it, that nitrogen, that protonated nitrogen can form ionic interaction with what in the DNA? مع الريبوز, deoxyribose, لا. Deoxyribose عليه hydroxyls, وهي الهيدروكسيل it's not ionized at physiological pH. pKa لها تقريبا 16. ايش في شيء بالدي ان اي؟ الفوسفيت جروب exactly. So the phosphatic group is, let's say, has a negative charge, and that part will be, أو الأكسجينز اللي على الفوسفات they have high electron density, بالتالي they can form, يعني تقريبا negatively charged, they can form ionic interactions with this nitrogen. Okay. طيب. That is why if you would remove the ribose part بالdoxorubicin, أو حتى كمان if I would, for instance, change that nitrogen, remove it or change it مثلا with a methyl, the expected activity راح تكون جدا جدا قليلة, or to a certain point the drug can be considered in that manner inactive. Okay. But here's an image of the DNA complex with 2 molecules of doxorubicin. Again, notice that the planar part, those 3 rings in here, are the ones that are intercalating between base pairs. With doxorubicin, زي الactinomycin D, it approaches the DNA from the minor group, or it binds in the minor group. لأنه بتكون خلينا نحكي البase pairs, the backbone, الهيليسز بتكون way closer to each other, which allows for much better interaction. If the drug is approaching the DNA from the major groove, راح تكون الother, let's say, أو خلينا نحكي those non-planar ring systems, they will not be able to approach سواء كان الباكبون of the strand أو حتى الnucleic bases حتى يعملوا good interactions معهم. Right? Here's a zoom in just to see that nitrogen of this glucose-like ring that is ionized. As you can see, it forms interactions with the base pairs. وبرضه كمان, it's not shown, يعني unfortunately crystal structure, it also forms ionic interaction with the oxygens of the phosphate in the backbone. Right? هلا الdoxorubicin, there are, يعني if you look at its structure, I would also expect إنه a natural compound. All right. Besides the doxorubicin, they were also successful in isolating closely related anthracyclines. يعني هاي الدراجز بسموها anthracyclines لأنه هاي الرينج سيستم تشبه الانثراسين. That is why it's called anthracyclines. هلا if you look at the structure of the other derivatives of doxorubicin, such as الإبيروبicin, donorubicin والإدارubicin, الvariations ما بينهم اللي هي the nature of the substituents على let's say this hydroxyl ketone, or this what we call an alpha hydroxy ketone group, أو حتى كمان على هاي الرينج أو الجلوكوز رينج ال R4 وال R3 وال R1. Okay. اللي بيختلف عنهم majorly, and it also affects the bioavailability والmetabolism اللي هو الإدارubicin. الإدارubicin المajor change إنه فيهم كلهم there is a methoxy group in على الplanar part. الإدارubicin, instead of the methoxy, we have a hydrogen. هلا how this increases or affects the pharmacokinetic properties of this drug versus the other derivatives of anthracyclines, إنه أنا عندي إذا بشيل الميثوكسي وببدلها بهيدروجين, how do you think the lipophilicity will be affected? بتزيد. هلا we have an oxygen instead of a methoxy واللي فيها oxygen, I've replaced it with a hydrogen. هلا الميثوكسي أو that oxygen, أو if I have a methoxy, that oxygen can form hydrogen bond interaction. إذا بشيلها تماما معناها إنه as if I am removing a functionality that can form hydrogen bonding. فالlipophilicity بتزيد. تمام؟ But because of that, because of the increase in the lipophilicity of idarubicin versus the rest of the anthracyclines, that is why cellular uptake بيكون أفضل. Another thing, أنا بجوز نسيت أنبهكم عليها من البداية. هلا أي سلايد أنا حاطة هيك فريم بلو عليه. This is an extra figure. بس عشان بدي أوضح again فكرة. مش مطلوبة منكم هاي الانفورميشن اللي أنا عم بشرحها هون. But just to show you why pharmacokinetics of idarubicin are enhanced, because if you look at the metabolism مثلا الdoxorubicin, one of the metabolism pathways اللي بتصير عليه إنه بصير في عندي since I have a methoxy here dealkylation أو dealkylation تنشال المثل بصير في عندي a hydroxyl that can be either glucurinated or sulfated which will highly increase the excretion of doxorubicin. But if you remove that methoxy معناها أنا عم بشيل هاي الجروب اللي ممكن يصير عليها conjugation وبتعمل elimination للدرug. So that is why the pharmacokinetic properties of idarubicin versus the doxorubicin, epirubicin, daunorubicin is much more enhanced because I have our say derivative the methoxy when removed الميتابوليزم اللي بيصير اللي بيعمل elimination للدرug شلته. Another thing lipophilicity right. طيب هلا what is known about doxorubicin إنه it stabilizes the DNA with topoisomerase 2. فمعلش تحملوني بدنا شوي هيك ندخل بتفاصيل what is topoisomerase, what it does, let's say where doxorubicin acts and so on. فكل هاي التفاصيل اللي راح نحكيها مش راح يعني مش مطلوبة منكم اللي راح at least it will help you to understand how مثلا doxorubicin when binds to topoisomerase 2 it inhibits it from doing its action. Okay, let me see first if you already have enough time. Yes. احنا لا وربع. طيب هلا during replication or transcription in both processes لازم يصير لازم ال DNA needs to be opened. What is the enzyme that opens the DNA? بما انه helix لا it's not topoisomerase. بما انه helix وانا عم بفتحها helicase. alright واكشلي doxorubicin في literature بيحكي او بالliterature you will find انه one of the enzymes that it inhibits اللي هو الهيليكيز. احنا ما بدنا نحكي عن هيليكيز احنا بدنا نحكي عن topoisomerase. هلا تخيلوا مثلا two ropes that are wrapped on each other. لو مسكتوا هالropes او هاي two let's say ropes اللي ملفوفين على بعض وفتحته شو بصير at the other end? Exactly it will be let's say complex. هاي الكومبلكسيشن اللي بصير هاي بسموها super coiling. alright هلا that super coiling mostly is alleviated by topoisomerase 1. هلا topoisomerase 2 اللي بنحكي عنه واللي هو mostly involved ب خلينا نحكي alleviating the stress اللي بيصير على DNA structure during replication. إنه during replication مش بتنفتح عند DNA strand أو helix. كل strand is used to build another helix. فeventually اللي راح يصير alright اللي هو those two newly أو اللي هم نسميهم إحنا two daughter DNA helices راح يكونوا شلبكين على بعض. In particular أو ال scientific term that is used for that اللي هو catenation. They will be coiled on top of each other. The enzyme that is responsible to alleviate high catenation اسمه topoisomerase 2. Okay? هلا what topoisomerase 1 does اللي بيعمله إنه when it binds to the supercoiled DNA as you can see in here what is that oval structure اللي has a blue color this represents اللي هو topoisomerase 1. اللي بيعمله topoisomerase 1 إنه بيرتبط في DNA strand أو أسف DNA helix بيكسر one of the strands just one. alright هلا بلف then it rotates where it broke the DNA it rotates it بحيث إنه it will alleviate the supercoiling بعدين شو بيعمل له religation. okay هلا topoisomerase 2 which as we have mentioned it alleviates the coiling or alleviates the catenation of DNA. يعني eventually when it replicates the DNA those two helices will be bound together هلا to alleviate that binding أو اللي إحنا بنسميه الcatenation. Okay, this is where topoisomerase 2 comes in. اللي بيعمل الtopoisomerase 2, which is represented by this orange colored structure, it binds to those, let's say, strands that are helices that are catenated. Okay, what it does, it breaks one of those helices من two strands, بيكسر two strands of one helix. Okay, then it changes their relative position, then religates it again, and this is how the coiling of the catenation is alleviated. يعني تخيلوا وكأنه مثلا أنا عندي two rings، يعني وكأنهم ال two DNAs, two rings, let's say bound together. What this enzyme does, it binds to where they are bound, بيكسر one of the helices من ال two strands، مش بكسر بس one strand, two strands، بكسر الهيلكس كاملة، بعدين بيحركهم عن بعض أو بيبعدهم عن بعض وبعدين بيعمل religation. Okay? So the difference between them, 1 و 2, إنه في حالة 1 it breaks one strand بالDNA, topoisomerase 2 breaks two of the strands، يعني الهيلكس كاملة بيكسرها. Okay? The image you see in here again, it summarizes what topoisomerase 1 and topoisomerase 2, الفاريشن ما بينهم. Alright? طيب هلا تتوقعوا هاي الكسر يكسر ايش؟ النيوكليك بيسز ولا الباك بون؟ الباك بون. برأيكم ايش اسهل اشي ينكسر في الباك بون؟ الرايبوز نفسها يكسرها ولا يكسر الباند ما بين الرايبوز والفوسفات؟ Yeah, it is covalent. حتى كمان الcovalent bonds can be broken. Alright? هلا how they can be broken, this is eventually يعني بالنهاية اللي بصير inside the active sites of enzymes, since in a topoisomerase 1 or 2 are actually enzymes, the process that occurs in is actually simply chemistry. Simple chemistry that we usually can replicate in lab. طبعا بشكل كثير أصعب. هلا let me explain even further. When topoisomerase 2 binds to the DNA where it needs to break it, سواء كان a single strand أو a double strand. يعني برضو كمان topoisomerase 1 eventually it acts in the same manner. هلا topoisomerase 2 and 1,2 they have in their active site an amino acid that has this side chain اللي اسمه ايش؟ tyrosine. لا التيروسين أنا بعتبره nucleophilic ولا electrophilic برأيكم؟ What do you think? Does it have any functionality that has high electron density? ليش لا؟ Yeah, that oxygen of tyrosine is considered nucleophilic. All right? هلا what you see in here represents what? Yeah, that's the DNA backbone. That's the deoxyribose bound to a phosphate, another deoxyribose, and the, let's say, chain goes on and on. All right? هلا the enzyme when it binds to the DNA بيحط exactly التيروسين وين الفوسفات موجودة. Exactly, فبقربهم جدا من بعض. هلا الفوسفات هون is electrophilic. ليه؟ لأنه أنا في عندي so many oxygens بيسحبوا electrons منها. البونز ما بين الفوسفات والأكسجين صح covalent بس still مش كثير قوية. يعني ممكن a nucleophile such as this oxygen can break it. All right? So what happens inside the active site when topoisomerase binds to the DNA, all right? The tyrosine in the active site, which is the catalytic amino acid, the oxygen will attack the phosphate أو بتكسر الباند ما بين الفوسفات والأكسجين of the deoxyribose, all right? And as you can see the result here, the bond, now the DNA strand is broken and the DNA is now bound to topoisomerase 2. Okay? طيب الريلايجيشن بتتوقع كيف بصير؟ يعني كيف برجع الانزيم وبرجع يعني بيشبك يعني بعد ما let's say rotates الهيلكس او حتى for instance يبعد هاي التو هيليسز عن بعض. كيف بيعمل religation بالDNA؟ And eventually the enzyme does not intend or is not, let's say, designed حتى انه يكسر DNA and keep it broken. Chemically كيف بصير religation؟ What was the bond broken? This one in here, right? لما احكي religation يعني رجعت الستراند حطت في بعض، او هاي that bond broken is to be formed again. فلو انطلب منكم مثلا ترسموا a mechanism for religation, what would you do in that matter? Exactly, that oxygen will attack the phosphate again, breaking the bond between the phosphate والoxygen of the tyrosine. هون برجع بعمل religation, and now the enzyme will be separated from the DNA. No more covalently bound. Okay? You don't have, and don't worry about understanding the exact mechanisms for topoisomerase 1 and 2. You just need, let's say, to have a somehow a basic understanding of what those two enzymes do, in particular how they do it. And eventually the breakage of DNA, سواء كانت a single strand or the two strands together along with religation, this is basically a chemical interaction happening inside the active site of the enzyme. Okay. طيب فقط معلومتين أخيرات بدي أحكيهم عن الدوكسيروبيسيل وبنكمل إن شاء الله الانتركليتنج agents next lecture. هلا recall إنه في الدوكسيروبيسيل structure حكينا إنه this planar part is the one that interacts with the DNA. Okay, intercalating between the base pairs. The nitrogen of the ribose part forms interaction with the ribose. Also, this part in here can form interactions with the DNA backbone or other nucleic acids. هلا لما نحكي إحنا إنه it specifically inhibits the topoisomerase 2, that's because this drug also, when it complexates with the DNA, it can also form very good interactions with topoisomerase 2 also. We'll stop here. المحاضرة الجاي بس a few information about what we mean by poison versus inhibitor. نحكي عن other mechanism for doxorubicin ونكمل intercalating agents. يعطيكم ألف عافية يا جماعة.